Hi all, let's wait for 5 more minutes and let's start the session. Hi all, welcome to the third week of Biophotonics NPTEL course, and I'll be your PA for Tuesday. I will introduce myself. I'm Sijit. I'm a research scholar of IIT Madras, and my working area is actually interdisciplinary between chemistry and biotechnology. And my broad area of study is actually unfolding and degradation of motor protein using optical pieces. So, yeah, hope the classes are good and you people are enjoying the classes. So let us start from the third module of our biophotonics course. It's a TA work, and yeah, hope you people are already gone through the course materials, and you can see that yeah, as per the course structure, you people are actually familiarized with some physics concept. Now, yeah, in this week, actually, whatever you, yeah, in this course, actually, what you are getting familiarized with your bio concept. So, let us start with the third module, which is actually the introductory biology part of our biophotonics course. So, I will, the structure will be as follows. I'll be solving some sample problems, which is given from NPTEL team itself. And what we will do, along with the problems, actually, we will try to understand more and more about the terms, which is actually included inside the problem. So, actually, each problems we will analyze in the problem aspect as well as the technical term aspects. Okay, so let's start. This is going to the first problem of the biophotonics course, which is regarding the basic building materials of our genetics. It's a nucleic acid. So the first question here given is nucleoside contains. So I have to understand what is a nucleoside and what it contains and how it different from other structure or something. So here the options which are given are A, base sugar, B, base phosphate, C, base sugar phosphate, D, sugar and phosphate. So before going to the actual uh, question and the actual answer of that particular question, let us see what are the two types of sugar present in our the basic building block, which is a DNA or RNA or something. So there are two types of sugar which is present in our chromosome or DNA, we can say. The term will come afterwards. So I am not reading now, such that the essence of the question will not go. So there are different. You can see that there are two types of sugar. Which is one is a ribose sugar and one is a deoxyribose sugar. So this here, what is the difference between here ribose sugar and deoxyribose sugar? You can see in the case of a ribose sugar, the second position carbon atom containing one H and one OH group, and this is bind to that ring-like structure to CC bond and CC bond. The other two valencies, actually filled by one is H and another is OH group. But in the case of a deoxyribose sugar, what happens? The OH group is missed there and it is containing only H. So this is the actually difference between one ribose sugar and a deoxyribose sugar. And let's see, when one oxygen is removed from that ribose sugar, that is the form. That's the name suggests that that's actually deoxyribose sugar. Oxygen removed ribose sugar. Okay, now, and this is the sugar part in our DNA or RNA or something. Now comes the nucleoside. This sugar, along with a particular nitrogen base, is called a nucleoside. So sugar plus a particular nitrogen base is called the nucleoside. So there are like different types of nucleosides are present here. So they are uridine. These are OH, which you can see. So this is actually ribose sugar. because there are two OH is here, OH is here, two OH is here, two OH is here. So all are ribose sugar. So it's a ribose sugar plus a nitrogen base. So this is called a uridine. This is a ribose sugar plus another nitrogen base. This is cytidine, and this is a ribose sugar plus another different nuclear, sorry, nitrogen base, and this is called a adenosine. And this is the ribose sugar plus another nucleotide is called guanosine. So these are the four nucleoside, and since it is a ribose sugar, so this is actually the building material of our ribonucleic acid or RNA. Similar way, in the case of a when the ribose sugar is different from, instead of ribose sugar, it's a deoxyribose sugar, it can called it deoxy, and the remaining name what is happened. Actually, it is in the ribose sugar, ribose, in the case of a nucleoside of a ribose sugar. But the thing you can see that here, in the case of deoxythymidine, here the nitrogen base is different that of a uridine. Actually, you can see there is no one extra methyl group here. All others are similar, but in the case of a deoxy sugar, the connected nitrogen base is different. Okay, you can call it as uracil, and this is called a thymine. In the case of cytidine, it's the same structure as cytidine. Only the difference is that ribose sugar is removed to form deoxyribose sugar. So it's a deoxycytidine. In the case of adenosine, it's a deoxyadenosine because ribose sugar is replaced with the deoxyribose sugar. In the case of guanosine, it's a deoxyguanosine. So it's a similar manner. Only the difference is that in the case of ribose sugar, it is uridine, and the deoxyribose sugar, it is a deoxythymidine. Okay, that's actually the building blocks which makes our DNA or the RNA structure. The top one is actually the RNA structure, and the bottom one is actually the DNA, the building blocks of DNA. Now see what happens. Whatever we see is like a there's a sugar, it's a ribose sugar or a deoxyribose sugar, and there's a nitrogen base. And along with the sugar and the nitrogen base, if there is the phosphate group is also attached, that is becomes the monomer of our the long polymer genetic material carrying molecule called DNA, or this the ribose sugar becomes a RNA. Okay, this is called this monomeric structure is called a nucleotide. The sugar plus nitrogen base is called nucleoside. The sugar, nuclear nitrogen base plus the phosphate group is all together is called a nucleotide. The nucleotide is the building of material of nucleic acid, which is the long chain genetic information carrying molecule. These are the different nitrogen bases present. One is adenine, and this is guanine. Another is cytosine, another is thymine. And you can see that this guanine, adenine can form two hydrogen bonding with the mm, nitrogen base, which is called thymine. Similar way, guanine can form a triple hydrogen bond with the other molecule cytosine. You can see that these are two ring structures, and these are single ring structure. And the two ring structures are actually called purines, and single ring structures are called pyrimidines. Okay, and you can see that in this one only two double bonded structure will form only double bond with the corresponding hmm, nitrogen bases. So we can see that this is a complementary basis. So adenine's complementary will be thymine, and thymine's complementary will be adenine. Similar way, guanine's complementary will be cytosine, and cytosine complementary will be guanine. So now let's comes to the answer. The nucleoside is a structural subunit of nucleic acid has hereby contain controlling components of olivine cell consists of a molecule of sugar linked to a nitrogen containing organic ring compound. So that's the answer. Answer the nucleoside that's actually containing the sugar and a nitrogen base. Okay. Now let's move to the second question. Bases which are present in RNA but not in DNA. So that we already discussed here. Bases which are present in RNA but not in DNA. What's DNA? DNA is a deoxyribonucleic acid. RNA is a ribonucleic acid. What's the difference between deoxyribonucleic acid and RNA? Ribonucleic acid, you can see here we already discussed that in the case of uridine, the nitrogen base here is actually uracil, but in the case of deoxythymidine, the nitrogen base is thymine. And what is it? What is the basic structure in DNA? Is deoxynucleotides. So the sugar is actually deoxyribose sugar. So deoxyribose sugar is only present in that in the case of DNA, but in the case of RNA, that is a ribose sugar along with the nitrogen base uracil. So RNA containing uracil, cytosine, adenosine, and guanosine. In the case of DNA, deoxythymine, it's called thymine. So DNA contains deoxythymine, deoxycytidine, it's called cytosine also, deoxyadenosine, and deoxyguanosine. Okay, now let's check the question. So present in RNA but not in DNA. Adenine, cytosine, guanine, thymine. These all are present in DNA. Arginine, cytosine, guanine, uracil. This is arginine. The cytosine, guanine, uracil are nitrogen bases, but arginine is a big amino acid. So this question, this will never fit into any of the molecule. It's adenine, uracil, cytosine, guanine. So yeah, we can see that adenine in DNA contains adenine, thymine, guanine, cytosine, but in the case of thymine, the uracil is replaced. So adenine, uracil, cytosine, guanine is the nitrogen bases present inside RNA but not in DNA. Adenine, cyanine, glutamine, thymine. This glutamine and is a amino acid. So that question also become an answer of the given options. and see the difference between thymine and uracil. So both are have the almost similar structure, except there is a one extra methyl group. In the case of a thymine, that methyl group is absent in the case of uracil. Let's discuss the another question. Just wait for some time. So let's go for our third question. Yeah. Which organism was used in Griffith's experiment to prove the genetic material can be transferred through a process called transformation? It's actually a nice question. From here, actually, we can understand a lot of things. And here, this is the experiment. There are two bacterial strains available. One is non-virulent and another is a virulent. What is called a non-virulent strain is called a rough strain because its cell wall is actually exposed and you can see the rough structure on the wall. The smooth strain is actually contains one extra layer of mm, extra layer such that it is actually it has a efficiency to overcome the antibodies present inside our organism. So there are two bacteria strain. One is rough strain because it's a rough structure of our cell wall is exposed. Another is a smooth strain. So it contains an extra layer such that this appears to be smooth, but that is actually a protective layer that has the capability of have the capacity to withstand against the antibodies inside our mm, cells. So what happens? These are two disease causing cells, and if you introduce this rough strain inside a organism, what happens? They use the mouse. They put a rough strain inside the organism. What happens? The mouse lives because the immunity, the immune system of our mouse will actually destroy the rough strain bacteria, and the organism or the mouse lives. What happens in the case of a smooth strain? In the case of a smooth strain, the smooth strain have an extra protective layer, and that will actually withstand against our immune system or the mouse immune system, such that the immune system cannot destroy the bacteria, such that the organism is affected, disease, and the mouse dies. What these people did in the case of Griffith's experiment, they know that this smooth strain bacteria are actually mm, smooth strain bacteria are actually uh, pathogenic, or it is causes the mouse to die. So they actually took the bacteria before introducing inside the mouse. They actually heated it, and the heated bacteria got destroyed, and they introduced inside the mouse, and they came to know that mouse sleeps. What's the meaning? Heat can actually destroy the smooth strain also, and if the destroyed smooth strain is actually getting inside the organism will survive, and it is not getting affected by the disease. So that also confirmed. What is our original experiment? And what they did, this the remaining things means the heated smooth strain, the heated smooth strain remnants, they actually mixed with the rough strain bacteria. The rough strain bacteria are actually non-pathogenic, and it will not cause any problem to the organism. So they mixed that rough strain hmm, organism with the remnants of the smooth strain organism heat killed, and after that they introduced the mixture to the organism and observed. What they observed was a very curious thing, such that they came to know that the organism died, or the mouse died. So what they observed was they found the presence of smooth strains inside the organism. So what is happening? So what is happening is when so this is a came to know that something is converted the rough strain organism to produce the extra protective layer against that. So how the extra protective layer is got produced, and how this rough strain bacteria became a smooth strain bacteria. So something from the heat killed smooth strain is transferred inside the rough strain bacteria, and the transferred thing was actually a something which is capable of producing the smooth, is converting the rough strain into smooth strain by producing the particular capsid, or the extra protective layer against the bacterial cell wall. And here one more thing we can see that, and that is actually thermally some more stable also. So this is actually a information that so later actually they found it that this is the DNA which is actually getting transferred from one bacteria to another bacteria, or from that bacteria remaining thing to another bacteria, which is a DNA which is a more stable structure, such that it, even though at the when the bacteria got destroyed by heating, that DNA remained unaffected and it is successfully transferred inside the rough strain bacteria, and it made it is capable of withstand against the immune system of the body. So, and now I will tell what is the disease they used. They used the disease of pneumonia. Actually, they introduced a bacteria which causes the pneumonia. So, from here, actually, you can understand that what will be the bacteria which they used. Cannot be Bacillus subtilis and Streptococcus pneumoniae, Staphylococcus aureus, and Streptococcus pyogenes. So, the pneumonia causing bacteria is Streptococcus pneumoniae, and they used this Streptococcus pneumoniae to check the genetic material transfer from environment to the bacteria. And this process is actually called a transformation process. So actually we are actually using this particular transformation technique to introduce our required genomes inside a bacteria, such that the bacteria can produce the proteins. So it's the thing, whatever that bacteria has the ability to take the genetic material, which is actually the DNA containing some genes corresponding to some proteins, and they can intake the gene, sorry, intake the DNA, and it can produce the protein. And if that is actually matching with the DNA, the taken DNA is actually matching with that of the bacteria, then they can produce the protein corresponding to that corresponding gene also. So, and this method actually we are utilizing for the production of different proteins. So, for example, just think that if we have a human protein, and and you cannot actually, I have a human protein and I want to synthesize in a large amount. What I will do, I will actually transfer the, this is actually how we are using this one. For example, I want a human insulin. Human insulin I cannot take from human body, so that will not be ethically good and other things. So what we will do, we will find out that it be this is a insulin protein. We will find what is the corresponding gene of insulin. There will be a particular DNA strands in which the insulin is caught. What we will do, we will make a particular circular DNA called plasmid, in which this particular gene is a part, and this particular plasmid is actually transferred, or is transformed, is transformed inside the bacterial cells, such that it will produce more and more, because this bacteria has the ability to reproduce, sorry, divide by themselves, and it will replicate by themselves, and it will produce a large amount within short time. So if you put this extra DNA inside the bacteria, while replicating the bacteria, the DNA is also getting replicated, and this there are like actually extra information like this plasmid have the capacity to replicate by themselves, and also it will produce a large amount of plasmids inside the bacterial cells, and the bacteria also have the capability of reproducing by almost like 20 minutes in the case of a E. coli. And what will happen is, without knowing itself, this protein coded gene is a foreign protein or something, the bacteria will produce more and more protein corresponding to that gene. But that gene is actually the gene of human, such that the bacteria is actually producing a large and large amount of human protein rather than bacterial protein. And we will lyse the cells, means we will rupture the cell wall of the bacteria and take it out, and actually we'll purify the protein and use it for our purposes. And yeah, the purpose of transferring the genetic materials from environment to the bacterial body is called transformation. Which cell organelle is mainly responsible for that? This is, yeah. Now we can discuss about the next question. Which organelle is mainly responsible for the process known as the translation? So there are So we already told that the genetic code which is encoded inside an organism is DNA, and the DNA is actually found in the form of chromatin inside the cells. In the case of a what you call the eukaryotes, it will be inside the nucleus. But in the case of prokaryotes, it will be like it will look like it does not have a particular compartment like structure, but yeah, it still will be in the form of the primitive form of a nucleus will be there in the case of bacteria and other things. But what is the purpose of this one? Actually, this containing like all the protein codes to be synthesized for that particular organism. So if the organism is getting metabolized, and just there are yeah, the nutritions inside the organism is getting metabolized, then there will be some enzymes are needed corresponding to that enzyme. There will be some code in that DNA structure, and corresponding the code is actually decoded and forms a particular amount of protein. And yeah, the protein formation from the DNA to the cell is called two steps: transcription and translation. And you see from where the transcriptions happens. So there are like a long, this is like a structure of a cell, and it has a big cell wall, and inside there is a nucleus, and inside the nucleus there are like a chromatin cell there. So in this chromatin actually you can see the DNA. And what happens in the case of a DNA, and you can see that there are like pores inside the nucleus through which the nucleus cytoplasm and inside portion of the nucleus is communicating. So what happens in the case of a transcription, the codes, the codes on the DNA of that particular organism is actually read, and there is a complementary base pair is formed, and the complementary base pair will be the form of an RNA, which is called a messenger RNA or mRNA, and the messenger RNA contains the same thing as that of the complementary portion of your DNA. And each three nucleo called bases actually corresponding to one amino acid, and this mRNA or the messenger RNAs pass through this nuclear pores and reach at the cytoplasm. And the cytoplasm, this thing has to be decoded and form the particular protein. And this decoding and combining the amino acid to form a particular protein is happening on the cell organelle, which is called the ribosome. So here we can understand which organelle is mainly responsible for the process known as translation. So there is no doubt that the organelle which is responsible for translation is ribosome. Answer is actually ribosome. Ribosome is intracellular structure made both RNA and protein, and it is a site of protein synthesis in the cell. So let's move to the question number 5. Hello, am I audible? You can use either microphone or you can type in the comment box if you have any difficulty for using microphone, such that I can confirm that you are hearing my voice. Okay. So yeah, I started from 6 o'clock, and yeah, here actually we are solving the model problems for week 3. So now we reached at the fifth problem. So anyway, I will go through the problem sets again, such that you can see, and if you have any doubt, you can ask. And this session also we will upload in that forum or that spreadsheet, such that you can access that. So I'm sharing my screen. So this is the question number 5. What is the nucleotide sequence of a start codon? So what is mean by the start codon? So for every DNA, as we told before, the protein is actually synthesized from the ribosome using the messenger RNA, and the messenger RNA is corresponding to a particular codon, which is actually each codon will be representing the code for a particular amino acid, and combining with all the amino acid where particular protein is got synthesized. But the DNA is a very long structure. So and all the codes are not corresponding to particular amino acid too. So you want the cells or the corresponding protein or the what it called the ribosome to understand that okay, this is the starting point of our protein synthesis. So there should be some code which is corresponding to that start codon, and there's only one single particular code which is actually the start codon. So and what is the what is actually the nucleotide sequence of the start codon? If you know, you can just type or you can tell also. C. C means AUG. Yeah, and AUG codon is for AUG code for what amino acid? Typing. AUG code for methionine. Okay, so here I put that some extra details also we will get. So this is actually it's a codon table. This is actually a graphical representation of the codons and corresponding amino acids. So it can easily understand which is actually the codes. And you can see there are like UUU, UUC, UUA, UUG, and corresponding amino acid is here. Am I audible? Your mic is on, I think. So I will, yeah. Anyway, after that, this session actually I will go through this one again, since that we can get the proper thing. So this is actually the codon table. So this codon table is just a graphical representation of the codons and the corresponding amino acids. These are, it will come like a different varieties, and you can see there are like there are letters like U, U, C. C stand for cytosine, A stand for adenine, G stand for guanine, and U. U stands for actually uracil, or instead of U, you can use the T, and that it can be thymine. And that is like instead of UUU, it can be UU TTT also. So UUC instead of TTC. Like that way you can put. So you can see that different codons are there, or you can see that different codons actually correspond to one particular protein itself. You can say UUU and UUC will code for the same amino acid, and UUA and UUG code for the same amino acid leucine. And here is a lot of codons which is actually corresponding to leucine. UUG, UUA, UUG stand for leucine also, and there are like CUU, CUC, CUA, CUG, all stand for leucine. Okay, and AUU, AUC, AUA stands for isoleucine, something like that. So here also similar way. Start from the 5 prime will be the center portion. GAA stand for E, aspartate, say aspartic acid, glutamic acid kind of thing. You can get from this codon table. So, but he can only see that there is only one start codon, which is actually AUG. It can be either AUG. In the case of actually just in the case of RNA, we can say AUG. And in the case of the DNA, we can say ATG. That will be methionine. And here it's a methionine. If there's a start codon, then there will be a stop codon also. And there are like different stop codon. UAA, it can be TAA, UAG, or UTAG, UGA, or TGA. These are all stop codons. If that particular protein containing a stop codon is present there, automatically what happens? The protein synthesis is stopped, and that will be the last codon for that particular protein. Okay, so start codon should be always methionine, and your stop codon can be anything. This actually this codon thing actually we can use if you are doing some uh, like reactions, or you want to do some particular protein, or and particular amino acid you want, but you don't want it. particular nucleotide. You can see you want to get the amino acid glucine, but you don't want the guanine guanidine inside that particular gene. So you can check. So you can introduce either UUA or UUG inside your DNA sequence. But if you don't want the guanine to be present there, you can use a UUA. That will actually decrease the content of guanine inside your gene. So like that way, corresponding to different requirement, there will be different changes in the codon as well as, but your start codon will be always start with a methionine corresponding codon, that will be AUG or ATG. This is a direct question. Who got the Nobel Prize for the work for CRISPR technology? So I think you can give the answer. Which option or who is the one person? Yes. So Jennifer Doudna is the person who got Nobel Prize in Chemistry for 2020. It's the portion which I took from the Nobel organization's website only.
Uh, हेलो सर।
Yeah.
सर, मेरा जो मैं अभी जो ये बायोफोटोनिक्स का जो कोर्स है ना,
Uh-huh.
तो उसमें जो लेक्चर्स होते हैं 6 to 8,
Okay.
सर ये जो है ना, ये मेरा दूसरा या पहला या दूसरा लेक्चर है।
Mm-hmm.
तो मैं मतलब और थोड़ा टाइमिंग कॉलेज से आते मतलब होता है ना, तो मैं भर नहीं पाता। तो मतलब मैं मतलब ऑफलाइन एग्जाम है अपनी, ये
Uh-huh.
अप्रैल में।
Okay.
सो या तो मैं उसके लिए कौन सा मतलब एक सिलेबस टाइप कोर्स या उससे ऐसा कुछ मतलब है सर कि जिससे मैं पढ़ के मतलब प्रिपरेशन करके मैं एग्जाम दे दूं, बिकॉज लेक्चर्स में मैं मतलब आऊंगा। अभी तो मैं ट्राई ऐसा करने वाला हूँ,
Mm-hmm.
मगर ये एक प्रॉब्लम है। तो क्या इसमें वेबसाइट में तो ऐसा कुछ मुझे स्पेसिफिकली नहीं, तो मैं मतलब कैसा मतलब आप मुझे बताइए ना सर ऐसा।
So you are actually going through the website, na? Means whether you are going through the lectures, because the lectures is always available in that manner. I can speak in Hindi, English or Hindi. My Hindi is
हाँ
not good,
हाँ
but I can speak.
हाँ।
Which language you want, Hindi or English? My Hindi will be bad Hindi, but if you want, I can talk.
नो सर, इंग्लिश इंग्लिश इज ओके मतलब।
Okay. So, are you going through this biophotonics lectures? These lectures, like one, two, like that way. These lectures anyway don't have any timing, na? And these lectures are not like very big, so small. In 1 week, actually, you can cover this all lectures. Whether you are covering that lectures,
ओके ओके सर, सर वी कैन ऑल्सो डाउनलोड दिस लेक्चर्स।
yeah, lecture downloading option is also available here, na?
ओके ओके सर, तो मतलब इफ मतलब एग्जाम एंड द पेपर ऑफ एग्जाम is I think that this lectures oriented, because
Yeah, most of
if
yeah.
we cover all lectures, so then we can get almost good marks.
Yes, yes, yeah. These lectures are something like that way. This your exam as well as the assignment. It's not only the lectures and offline exam. Okay, you are solving assignment and submitting.
Yes, sir. I completed my 3 weeks assignments,
हाँ
and then this 4 weeks जो assignment अभी आया है
हाँ हाँ.
वो
So
हाँ, so this 4 week assignment I will complete within 1 or 2 days.
that's
So
okay. But yeah, that doesn't matter. But the thing is that yeah, along with your end semester exam or end semester of the offline exam itself, your assignment marks also will be counted. So don't forget to submit before the deadline. So and this is
uh,
the yeah, and you have to always what do you say? Yeah, you have to always submit that one before deadline, such that these are like a computer assisted thing. So nobody cannot do anything. If it's a deadline is crossed, automatically your submission is locked, and nobody cannot do anything like that. If you want extra time, nothing will happen there. And if you have any doubt regarding something, sometimes it's like these people are actually lot of questions and answers are making. Sometimes some mistakes can come, but yeah, if some mistakes you have doubt that whether any mistakes there in some problems or something, that you can directly share with us when we are taking these live sessions, or you can actually put it in that our there's a Google group now. In that Google group you can put it as a query kind of thing. So somebody will see that one, and we will actually, if there's a proper question which is actually some wrong, or which is actually making some confusions are there, actually we can remove the question also, and we will only the proper questions are getting graded, such that there will not be any problem with your marking scheme and other things.
yeah, sir. Uh, and sir, my एक दूसरी एक query थी, another query.
You can
मतलब
ask.
sir, मतलब I'm doing graduation in physics
Okay.
in Gujarat, Ahmedabad.
Mhm.
So sir, I think that I am मतलब I wish to मतलब get in IITs, one of the best IITs like Kharagpur, Delhi, Mumbai, Madras. That so if मतलब, this kind of you know uh, this kind of programs is I think very helpful to me. So
Yeah, and you can see that actually you can see that now this all the programs are like interdisciplinary in nature. So I am actually a graduate in physics only. Now I am doing research in this kind of thing. So my research topic is chemistry and biotechnology. So I am actually working in protein as well as working with the laser to manipulate another thing. That thing like optical tube something will come in this course itself. How these are working and how we will manipulate and all of the other things will come in this one. So I am a graduate in physics. Now I am doing my research in biology, biotechnology as well as chemistry. And these all facilities are like available in different IITs as well as now. Actually, people are doing more. Before it is like when it is physics, then all the core physics topics are will be the research topics. Now it is not like that way. In every branch, you can see something related to bio as well as chemistry, or also like a multidisciplinary kind of research is going on now. So these things will be available, and yeah, it is like if you have a specific interest, something like if you
yeah,
want to
so,
go into this bio field, so this kind of
मतलब,
course will be very helpful.
yeah. So in Gujarat, Ahmedabad, um, I'm मतलब my college is associated with Gujarat University. So in my university, in my college, the the environment is very rigid. मतलब,
Hmm.
because I am a graduate student in physics, so then I cannot doing research and go anywhere to a participant with research programs. They should. They said that you must completed your masters and PhD, then you get into a research. So I think that if IIT can approve me as a graduation degree, on the graduation degree, so that I can go there and doing research, and then after I come to Gujarat, Ahmedabad, and then clear masters and then PhD, and then okay in any government institute like IITs and PRL, and then I can doing my research job,
Yeah, that I
which is my
understood.
dream job.
But yeah, that there is another issue. Will be there like that way. Graduation means you are doing your BSc in Gujarat medium.
Physics.
Yeah. So when you are doing BSc, all IITs actually the graduate course actually starting with the either like BSMS or BTech, normal graduation. I'm not saying about online degree program. Online degree program is actually, I don't think this biophysics is not at all introduced here.
Yeah,
But yeah, all the online degree program is like now it's whatever they're giving, it will be like artificial intelligence, data sciences, electronic sciences, and that kind of things. Not like a direct what you call BSc degrees are not given by IITs till now. So, okay, yeah. So there is not a direct BSc program is available in IITs. And but if you join some program, actually you can do all the things. For actually to do the graduation, actually either you have to come through this JE advanced or something. Then only you can get into IITs for studying this one. Or the thing is that you can do masters. That's after your BSc. Actually, you can apply for masters, and during the masters, actually you can do all the projects and other things. And if you are actually, I don't know how the university structure is going on there, but if you are getting a proper vacation, then actually you can do the summer research fellowship program and all other things in IIT itself. So you may have to give some proposal, you have to give your
yeah,
willingness to your some professors where you are interested in, and you can actually
yeah. Just
contact.
doing a master's in so now I think that first of all, thank you so much to give me this some this kind of very important information, because I search Google, but Google is not मतलब उसमें समझ में नहीं आता है वो गूगल में वो क्या बोल मतलब सर्च इंजन में उसमें आंसर्स नहीं आ पाता for IITs, and then so
So if you want to do masters in IITs, actually you have to write the JAM exam, joint admission test for MSc. JAM exam will be there. uh,
okay, okay, sir. And then I think that I'm doing a master's in a Gujarat University, and then I am applied for a PhD because I like research more, because master's is matlab, one kind of steps in career. So for PhD, I am applying to IIT. So that is okay for me to matlab,
that is also okay, but just yeah, just think that I will tell that whenever you are, yeah, this is like, yeah, anyway, there is a little bit diversion, but yeah, this will be good for all the people also, I think. Yeah, anyway, we'll cover that all question also. So yeah, I'm telling that whenever you are doing something, your next target will be always somewhere higher than that one. Okay. So if you are doing BSc, and if your target of doing graduation or the MSc in IIT, your PhD will be, your target will be PhD will be some more higher institutes. So it's like a I can say that I am actually studying in IIT Madras. Yeah, whoever is actually graduating MSc here. So actually, the next target of them is actually they are doing PhD in abroad, not only in India. They are going either US or Europe or some other big institutes around the world. They are going. So it's like a, so yeah, always you will think about some higher. If you're thinking about doing masters in state university, actually you people will think about a degree doing in IITs. So after completion of so degree, I mean PhD. After completion of the PhD, you people think about doing postdoc abroad. So this will go down. So you can say that people are studying BTech in IIT. The next time, if they want to do MTech or they want to do MS or they want to do some research for this completion of the BTech, that these people are going abroad and doing higher education in topmost colleges around the world, not in India. So it's like a, always, if you are doing achieving your goal very fast, the next step will be like always higher. So actually, you can try for masters also, and along that one, if you are getting, let it be. If you are couldn't crack the exam, you can do your graduation there itself, and you can come to for PhD. So for getting PhD admission, there is a more criteria is there. So it is like you have to clear either gate or this is CSIR, JRF, net or something. So like that channel only you have to get into PhD admission, not from JAM. JAM is actually for doing MSc, and gate, net, and this CSIR, JRF kind of thing is actually for doing PhD.
And सर वो जो मतलब in IIT JAM, so in मतलब what kind of preparation and some kind of preparation and materials and IIT level material, IIT level मतलब like uh, uh, some like physics. So then फिजिक्स के लिए जो आईआईटी मास्टर्स का फिजिक्स है, तो उसके लिए आईआईटी जैम में मतलब कैसे मतलब प्रिपरेशन, बिकॉज गूगल पे बहुत सब मेस्ड मतलब है, कुछ पता नहीं चलता.
Okay,
एंड
let's do one thing. Okay, let that this session ends. Then other such that other people are also be here.
या
So,
या
so what I will do? Okay, after this session, you can talk to me. Okay,
या या
let this questions complete. After that, I can give whatever I know.
या थैंक यू सर.
Okay. So let's complete our session. So yeah, this is the Jennifer A. Doudna. She got the Nobel Prize for this CRISPR-Cas9 technology. Which actually give the, it's actually genetic scissor. Which actually you can cut a portion of your DNA and put something such that it will be like a very useful, and it's like a very big leap in the molecular biology studies. Okay. Now, what is the connective tissue in human body? This doesn't mean that is only connective tissue in the human body is the given below. So actually, you can have the question is like that: what is the connective tissue in human bodies? Out of the given options, what is the given connective tissue? What is the connective tissue in the human body? Is the question. So you can see neurons are there, blood is there, epithelial cells are there, muscle tissue is there. So what is the answer? If you know the answer, you can type it in your chat box. Let's settle the answer. Here, let's see the definition of the connective tissue. And here, actually, I'm giving a disclaimer that for this presentation, actually, I took a lot of images from Google directly. So yeah, I don't have any ownership for any of the image here. And I'm saying that this all the ownership of that image is actually belongs to that corresponding proper authors only. So this is only for education purposes. I that use this all images for this presentation. So I don't have any ownership for this images. So this is the connective tissue. This actually what they will do. They will, there are like a tissues which you use it to bind structures together. to form a framework or support the organism organs like that way. And it's also used to transport one substance from one place to another like that way. So the types of connective tissue which is actually present in our bodies are like adipose tissues, then fibrous connective tissues, elastic connective tissues, cartilage, osseous tissue, it's a bone, and blood. These are the different types of connective tissues which is present inside our body. This cartilage tissues, bone tissues, and this fibrous connective tissue, elastic connective tissues, the adipose tissues. These are like actually the connective tissues. And out of the connective tissues nature, different types of connective tissues, the options which is available here is actually the blood. So the answer here is actually blood. Now the question 8. The question 8 actually is asking which disease is that cause for protein misfolding. So which disease is happening because of the protein misfolding? There are four options are there. One is prion, another is Alzheimer's, another is Parkinson, D, all of them. Can you give any answer? Okay, D. So the answer is actually all of them. So here actually, yeah, these are like some interesting fact is there. Even though these are like, yeah, we are talking about disease. These diseases are actually coming because of the protein misfolding. We already told that there are actually proteins. How the proteins are getting synthesized? These are synthesized from our DNA. So from the DNA, the corresponding code is actually decoded, and some mRNA strand is made, and this mRNA strand and this formation is called a transcription. The mRNA strand is actually decoded using ribosome, and from the ribosome actually what happens, an mRNA strand, the code is actually read by the ribosome, and correspondingly amino acid is formed, and amino acid is actually present in the cytoplasm. The amino acid is combined to form this polypeptide bond, and the protein is actually getting synthesized from the amino acid. There will be like amino acid will be presenting in the cytoplasm. From here, from corresponding the code, the amino acid chain is formed, starting from methionine, and the protein chain is got formed. And the we already told that yeah, protein containing different structure, and this there will be some interaction, intra molecular interactions, and there will be like a ionic bond and salt bridge formation, something like that way, hydrogen bond, all because of this bond, the protein from the translated protein is actually folded to form some particular structure. And that particular structure is actually, it is not like a random structure, it has a particular structure which is corresponding to its function. So every protein having particular nature should have the corresponding proper structure. Otherwise, the function of the protein is getting affected. So if the protein have to do particular activity, it should the structure should be maintained properly. But yeah, so how the protein fold is actually a big question in our molecular biology. But the thing is that your people already see that there is a central dogma which is written there. So all the proteins also, all the coded information is there in the DNA. From the DNA code itself is actually telling the sequence to fold in a proper manner such that the protein synthesized will be always in a particular shape. But due to some reason, there can be a reason which is affected by some external environment or some random factors such that the protein may not fold in a proper manner. So what happens is that the protein should be folded in this way, and if instead of folding in that way. One protein is folding something like that way. So if it is fold something different than the original shape, this protein either will not function at all or it can do some harmful to the organism. So similar way, there are diseases that is assigned because of this protein misfolding. These diseases like Alzheimer's and Parkinson's, these are actually coming from the protein misfolding. The protein, instead of folding in a proper manner, it is folding some different manner such that it is getting misfolded. And this is something like a prion. Prion disease is actually a disease. It's a neurodegenerative disease actually, which is affecting our neural system such that and there's another problem. This for this prion protein. This prion protein is actually a transmissible protein, a disease. So you can see that this is like a one protein, let it be okay, this is a misfolded. So in cell actually there are mechanisms. So actually it can convert this to again back to, there are some another systems inside cell such that it can convert back to in a correctly folded type, or it can, what they can do, they can destroy the protein such that it will not do any harmful to the body. There are like different mechanisms available in the body to avoid this protein misfolding accumulation inside the body. But sometimes this thing will fail. So the protein which is misfolded will accumulate and it can cause a disease. But in the case of the prion protein, there's a one more problematic thing. So just see that this is a normal protein. A lot of normal proteins are synthesized. And due to some reason, this prion protein is something like it is misfolded. This misfolded protein is so dangerous such that it can actually induce this misfolding to all other normal proteins. In the case of other proteins, what happens? Whatever is misfolded protein will stay there, and if it is causing any problem, it can cause or it will be inactive. But in the case of a prion protein, what it will do? This misfolded protein will induce misfolding in all other proteins. So if some misfolded protein is there, then the good protein also will get misfolded. So what it will do? So if the protein is formed inside your body, it will also convert all your good protein also to be misfolded, and it will actually increase the severity of the disease. And this is actually a transmissible disease such that it can transfer from one organism to another organisms using this body fluids. So this is actually a mischievous protein, you can say, this prion. So this is the answer. The answer is a protein misfolding means to fold into an incorrect three-dimensional shape that's typically non-functional and often resistant to breakdown. These three Ds are related to protein misfolding, thus creating abnormalities. Okay. Then the receptor of coronavirus to enter the host cell. Which is answer? The answer is written like A. Where the receptor of coronavirus uses to enter the host cell. So see, this coronavirus will have a spike protein. And yeah, this question is I think a little bit confusing. I have one doubt I can ask. How you said the A something? Whether any doubt regarding ACE2 with respect to a Srivatsan? Whether I had to get it clarified from the higher people. If you have any doubt in that one, so this is like a, yeah, it can be A if that spike protein is also called a receptor. So what is happening? This spike protein is actually getting attached to the ACE2 receptor of the host cell, and then it is getting activated using TMPRSS2, and this spike protein is got cleaved, and this genetic material from this virus is actually loaded inside the host cell. This is the way the coronavirus is actually getting affected our system. So if the spike protein is also a receptor, then the answer is actually A. But the receptor is actually present inside the host cell, and if they are asking about the host cell receptor, then the answer can be ACE2, or they are asking about the attachment protein as a receptor, then the answer can be our spike protein. Okay. So if you have confusion, I can contact this higher authority whether if it's have a confusion, or if that is actually whether the spike protein is also called a receptor. That's my doubt. I am also not completely from. I am getting into that biology field in a proper manner only. So I cannot author with authority. I cannot tell that spike protein is also receptor. Whether spike protein is also receptor, or it is called a receptor also. That I have doubted. So that's why I am not saying. So I can clarify that one in the next class. But yeah, if receptor is only telling about the host cell, then the answer can be ACE2. But it's telling about the structure on the coronavirus, then the receptor is not present there. The structure which is actually helping the coronavirus to attach the host cell is a spike protein. What is the function of lipids in cellular structure? What function the lipid will do? So it's a direct question. It's- It gives structural stability, it stores energy for cellular metabolism, both A and B, none of that. Sure. Yeah, that they can get clarified whether that is a tricky question, or that is a some while setting the question became tricky. I can ask. So let's see the answer. It gives structural stability. It stores energy for cellular metabolism. The lipid system actually contains a fat. Fat is actually a lot of. It's actually a good source of energy also. Okay. Lipids are a diverse group of molecules that share the characteristic that all at least a portion of them is hydrophobic and another part is hydrophilic. With the characteristic lipid, it can give structural stability. So actually it will form like a vesicle kind of thing. So one part is hydrophobic and another part is hydrophilic, and if the lipids are actually, you put it inside a hydrophilic system like a some water based system, what will happen, this hydrophobic form, hydrophobic entities will go to the core form, and the hydrophilic end will be at the surface, and it will form a globular kind of thing. And this kind of structure actually will give a structural stability. And and this is actually energy storing components and many more. Okay, so this is can, and lipids containing a lot of energy content also. Okay, so this is the total questions. So I think you people didn't see that first five questions now, so I can go through that first five questions also. This is the first question. Nucleoside contains a base sugar, a base phosphate, base sugar, phosphate, base sugar and phosphate, sugar and phosphate. What is the answer? Nucleoside. Nucleoside contains base sugar, base phosphate, base sugar and phosphate, sugar and phosphate. So you can give the answer. Don't have answers. So these are actually some. This is a two sugar which is present in the our nucleotide. So one is ribose sugar, one is deoxyribose sugar. Difference between deoxyribose sugar and ribose sugar is in the ribose sugar, the second carbon atom will not have a oxygen. Instead of OH bond, that will have a single H bond. It will be COH bond, but here it's a CH bond. So it is looking like one oxygen is missing. So instead of a, from the ribose sugar, one oxygen is mixing. So it's a without oxygen ribose sugar. So that's actually deoxyribose sugar. And this ribose sugar or deoxyribose sugar plus a particular nitrogen base is called nucleoside. So the answer will be base and sugar, that will become nucleoside. Okay, so these are like a different types of nucleosides. So it's uridine, cytidine, adenosine, guanosine. These are the nucleosides with the ribose sugar. When it becomes a deoxyribose sugar, it will become deoxycytidine, deoxyadenosine, deoxyguanosine, and deoxythymidine. There is a difference when it is become a deoxyribose sugar. The nitrogen base is not uracil; it will become a thymine. So, and this is the sugar plus a nitrogen base plus a phosphate group. This is actually the molecule which is a monomer of the long nucleic acid, and this structure is actually called nucleotide. The other sugar plus nitrogen base is called nucleoside. Sugar, nitrogen base plus phosphate is called nucleotide. Okay, these are different nitrogen bases. There are nitrogen bases which is containing two ring structure as well as single ring structure. And two ring structure nitrogen bases are called purines, and the two purines are adenine and guanine. And single ring nitrogen structures, nitrogen base structures are called pyrimidines, and they are cytosine and thymine. And you can see that adenine can make two double bond with the thymine, and vice versa. Similarly, guanine can make a double three hydrogen bond with the cytosine, and vice versa. And it can combine only in this manner. So what happens? So these are like hydrogen bonds, two hydrogen bonds or three hydrogen bonds. So this all things will stay in a plane only, and it will actually give a ladder like structure. So if you see, if these things are coming something like that way, this will be always in a plane. So it will give a ladder like structure, then it cannot do anything, it cannot twist or something. So only it can do is thing, it can just form like helical structure with the preserving the plane, something like that. And there will be some hydrogen bond between them. So this is the way DNA coils. Okay. Bases which are present in RNA but not in DNA. Some bases are present in RNA but not in DNA. Which bases are present in RNA but not in DNA? So this actually have all the answers. The question is something tricky. Which bases are present in RNA? So first answer, adenine, cytosine, guanine, thymine. So adenine, cytosine, guanine, thymine. These are the bases present in DNA, not in RNA. And this arginine, cytosine, guanine, uracil. And this is a tricky. This arginine is actually a amino acid, not at all a base. So this question is not at all belongs to any of these questions. It is not at all present in RNA also, and it's not present in DNA also. Another is adenine, uracil, cytosine, guanine. So here actually that thymine became uracil. Adenine, guanine, cytosine, uracil. So this is the nitrogen bases present in RNA. Adenine, cyanine, glutamine, thymine. And this glutamine is actually a amino acid, not a nitrogen base. Again, this is not present in any of the nucleic acid. So the answer is C. And see, this is the difference between thymine and uracil. You can see all structure is similar, but this thymine have an extra CH3 bond. One single line is actually corresponding to one single line means actually CH3, one methyl group. So in the case of thymine, there's one extra methyl group is there. In the case of uracil, that methyl group is actually absent. Which organism was used in Griffin's experiment to prove that genetic material can be transferred through a process called transformation? So this is the experiment in which what they did, there are like two strains of a particular disease. Now I am not saying the disease name. There are two strain of a particular disease. One is rough strain because the cell wall is actually rough and it is exposed. There's a smooth strain. So this containing some extra protective layer around the cell wall such that it appears to be smooth, and it's actually immune against the immune system of the organism. So if the rough strain is actually introduced inside the mice or the organism, that immune system of the mice is actually destroying the rough strain, and that mouse lives and this all bacteria dies. In the case of smooth strain, it has an extra protective layer, so mouse get died. What they did, they actually heat killed the smooth strain and then introduced to the mouse, and the mouse survived. And what they did, the heat killed the smooth strain remnants they mixed with the rough strain, and they introduced it into the mouse. And they found that the mouse died. And when they opened that, they see that mouse remnants, they found that there are like a smooth strains is present there. So some rough strain became smooth strain, or something from the smooth strain is actually transferred inside the rough strain bacteria and made the rough strain bacteria to act like a smooth strain bacteria. And later it is discovered that this transferred thing is actually the genetic material, and this is nothing but DNA. And the transformation, and this particular process is actually called a transformation, which is a cell organ mainly responsible for the process known as translation. So translation is the process in which from the DNA sequence actually protein is getting synthesized. And this is happens at a two step. One is the from the DNA a messenger RNA or the mRNA is formed, which is containing the complementary code for the DNA strand. And the messenger RNA is actually transported from nucleus to cytoplasm through this nuclear pores and it's reached at the ribosome. From there, the code is decoded and the corresponding codons, amino acid is collected and it is combined to form a protein. And the transcription, the formation of the messenger RNA from the DNA is called a transcription, and the decoding of the messenger RNA to form the protein translation is called translation, and it is happening outside the nucleus and it is happening at the ribosome. So the answer is actually ribosome. I think from here actually questions you people are already covered. Hope this session was useful and understandable for all. Thank you. Let us see. Let's meet next week.